Hello, and welcome to this video on Community Profile Analysis as made for MIMG 109BL at UCLA. This first video is on the OTU table. In this video, we will review the methodologies performed for cultivation-independent microbial ecology research. By the end of this video, the student should understand the process from metagenomic DNA extraction to the construction of a community profile, and then should be able to perform basic community profile analysis with Excel. The methodologies for cultivation-independent research start off with soil collection. In winter of 2017, the 109 class collected soil from the Sedgwick Reserve in Santa Barbara, looking closely at the characteristic serpentine soil within the area. The next step is to perform a metagenomic DNA extraction using the Microbiome Power Soil DNA Isolation Kit. Take a moment to review how this kit works. The Microbiome Power Soil DNA Isolation Kit comes with 6 reagents. The first, C1, lyses the bacterial cells, releasing the DNA into the environment. C2 and C3 precipitate out the proteins, the cellular debris, and other impurities from the soil sample, while C4 is the aqueous high salt solution that binds the DNA in the soil sample to the spin filter. While the DNA is bound to the spin filter, C5 washes out any non-DNA particles from the sample. Once that has been performed, C6 allows the DNA to be eluted from the membrane and allows it to pass through the spin filter, thus releasing the bulk DNA of your soil sample. Our bulk DNA was then sent off for 16S rRNA gene-targeted next-generation sequencing by Illumina, and this involves the clonal amplification of the 16S rRNA gene fragments, as well as sequencing by synthesis. And this is a process in which fluorescently tagged nucleotides are added to the strand being read. This is done on a massively parallel fashion, as represented by this picture where one dot represents the addition of one nucleotide to one strand on one sequencing platform. If you want to review the steps of next-generation sequencing in more detail, Illumina has some videos available on YouTube. Very much like Sanger sequencing, the outputs of high-throughput sequencing lack biological information, and this is where CHIME comes in. CHIME stands for Quantitative Insights into Microbial Ecology, and it is a bioinformatics package that transforms raw sequencing data into text files that can quantify operational taxonomic units, or OTUs. These OTU tables can then be used for biostatistical analysis. And this is what an OTU table looks like. Take a moment and make some observations. First, you'll notice that the taxonomic information is presented in a hierarchy to the left, and this means that each level of taxonomic classification is defined in separate columns. You'll also notice that these taxons are quantified with these OTU counts for each sample community towards the right. Some OTU tables also have the quality scores for taxonomic assignment similarly to the outputs of a BLAST search. Because all of this information is available in Excel, now you can perform some community profile analysis. You can use preset functions such as equals some values and equals average values in order to explore your data. You can also define your own formulas using the basic computational science, and this may be helpful in calculating the Shannon Diversity Index or any other index of diversity you choose. Don't forget that Excel also has tools for data visualization, such as pie charts, bar charts, and heat maps using conditional formatting. Before I conclude this video, I leave you with some observations and considerations. Consider how this data could either support or refute your initial hypotheses concerning your cultivation-independent research. Consider what graphs or plots are most appropriate to present your analysis. Also, What current observations could be used to formulate statistical hypotheses that could be tested later? Also know that because you are a UCLA student and you pay tuition, you get access to lynda.org's in-depth tutorials using Excel. All you need to do is log in using your BOL account. Thank you.